Human cloning. Let us first discuss what is cloning. Cloning is the process of making an identical organism through non-sexual means. In 1997, Ian Wilmot at the Roslin Institute in Edinburgh, Scotland, successfully cloned the first mammal, a sheep named Dolly. It is also known as somatic cell nuclear transfer, the technical process by which cloning is performed. Examples of cloning. Human cloning. It is the creation of genetically identical copy or clone of a human. The term is generally used to refer to artificial human cloning, which is the reproduction of human cells and tissues. It does not refer to the natural conception and delivery of identical twins. Monozygotic or identical twins are natural clones. Types of artificial human cloning. Reproductive cloning and therapeutic cloning. Reproductive cloning involves production of genetically identical individuals. Therapeutic cloning, used to produce embryonic stem cells in order to repair damaged tissues and organs, involves cloning cells from a human for use in medicine and transplant. The biggest misconception the general public has about this research is that cloned individuals will not be the same person as his or her donor. Although the donor and the clone will look exactly the same physically as they have identical genomes, internally they would be different individuals. The environment in which we have grown up in and the experiences that we have throughout our life defines us and shape us uniquely as individuals. So as Wayne Traub writes, we are all more than the genetic instruction manual that has involved in our creation. Even the nature's clone or the identical twins are not identical. They have the same personality, character, interest, styles, etc. They grow up to be different individuals. So let us first know who was Dolly. Dolly was a Finn Dorset sheep that was born in 1996. Which makes her so special is that she was the first clone of an adult mammal using adult cell cloning methods. So the ethical issues on human cloning, the technical and medical safety. So the technically human genes grows older with age. It's feared that the cloned individuals would retain the age of donors' genes. Cloning animals through somatic nuclear transfer is simply inefficient. The success rate ranges from 0.1% to 3%, which means that every 1,000 tries, only 30 clones are made, undermining the concept of reproduction and family. Ambiguous relations of a cloned child with the progenitor, confusing personal identity and harming the psychological development of a clone. The ambiguous kinship could lead to psychological and emotional confusion for all family members. A woman can be the twin sister of her mother, lack a biological father, and be the daughter of her grandfather. In vitro fertilization has already led to confusion of parentage, but cloning will mean the radical rupture of these bonds. Human cloning must also be judged negative with regard to the dignity of the person cloned, who enters the world by virtue of being the copy of another being. This practice paves way to the clone's radical suffering, for his psychic identity is jeopardized by the real or even by the merely virtual presence of his other. Since the clone was produced because he resembles someone who was worthwhile cloning, he will be object of no less faithful expectations and attentions, which will constitute to a true and proper attack on his personal subjectivity. Concerns about eugenics and promoting trends towards designer babies and human enhancement. So eugenic is the selection of desired heritable characteristics in order to improve future generations, typically in reference to humans. Concerns about eugenics, the once popular notion that human species could be improved through selection of individual possessing desired traits, also have surfacing since cloning could be used to breed better humans. thus violating principles of human dignity, freedom, and equality. So it is contrary to human dignity. Consider the manufacture and destruction of human's life even though life has not developed past the embryonic stage. So despite several highly publicized claims, human cloning still appears to be fiction. There currently is no solid scientific evidence that anyone has cloned human embryos.